Hey, Mr. P here.
What's up, guys? It's Mr. Schmitz.
And in this video, we're talking about mitosis.
Hey, Mr. Pfeiffer.
Yeah.
What did one cell say to the other when they dropped a beaker on their foot?
I don't know.
Ouch! Mitosis.
Got any more?
No,
Oh, that's it.
that's it.
So, you're not going to have a joke for class?
I ran out of all of my jokes.
Yeah.
Hope
Mitosis.
Hope they watched the lecture.
Yeah. So, there you go. Bonus joke right off the bat. Mitosis is a follow-up video to our last video, which is the cell cycle. And in that video, we talked about all the phases of mitosis. So, specifically interphase, G1S and G2. And then we talked about the mitotic phase or M phase. And so in this video, we're going to just focus on that little tiny sliver of the cell cycle, which is mitosis, which is when the cell is actually dividing its nucleus.
So within mitosis, at this stage, there was interphase, right? It was G1, S, and G2. So moving from interphase into mitosis, have the understanding that we have duplicated our DNA, that we have grown and duplicated our proteins and organelles, and that the cell is ready to divide. So at that point, we're going to move through 4 phases, as well as ending with cytokinesis, that first phase being prophase, metaphase, anaphase, and there's some debate on how to pronounce this last one. I say telophase.
Yep, could be telophase.
Depends on who you ask. But telophase or telophase is the last stage of mitosis, and then it is followed by cytokinesis, which is technically its own process, but oftentimes gets included right here at the end of mitosis. So a good little way to remember that set of 4 phases is PMAT, which Mr. Pfeiffer went and highlighted the beginning 4 letters, PMAT. You can come up with a fun little way to remember that. But
Yep, people, meet, and talk. If you kind of like that, if you're into that kind of thing. Some people don't like to meet and talk.
pessimistic mice ate tacos.
Yeah,
Well, whatever you got to do to remember it.
right? You got to get that visual.
Um,
PMAT. You might even think of just a PMAT.
that's gross.
Yeah, probably don't visualize that.
I know you're training a new dog to go to the bathroom.
Yeah,
but...
so PMAT is on the mind.
Mm-hmm. So let's start with prophase. Pro. What does the prefix pro mean?
So pro means before. So this is going to be the first phase, or the thing that kind of happens before all of the mitotic, or all of the intricacies of the mitotic phase.
So let's touch on a couple of important things that are going to happen. The first is going to be that our nuclear envelope is going to dissolve. So you can see that exhibited by the dotted or dashed line there around the nucleus. Basically, we have to get rid of the nucleus because we got to get into the DNA and separate it out. And so the first thing that has to happen is get rid of the nucleus.
Yeah, and we have to get into that nucleus in order to manipulate the chromosomes. So we're not just dividing the nucleus. We're not just pinching it in half and dividing. We have to actually physically manipulate the chromosomes and kind of move them to different sides, which we'll get to as we progress.
So all of this is going to kind of be happening at once. We're obviously going to put it up one at a time. But the next thing that we're going to talk about is the chromatin condensing into chromosomes. So we're going from that unorganized sort of blob of DNA that exists during interphase, which is called chromatin.
And remember, that's the inactive form, right? And so in this diagram, it's the dots. And I know DNA is not just a bunch of individual dots. So this model is flawed, but the X shapes are the chromosomes, and they're being condensed from these dots, which would refer to as the chromatin. They produce the chromosomes as well. Centrioles are going to migrate to the poles, so these centrioles are these orange structures, which are going to not only be producing the spindle fibers, but they're going to be physically moving. So they will start kind of together, and they will be moving at the same time that they're producing these blue spindle fibers to the individual poles. So one's going to kind of terminate here and the other's going to terminate here, so that we can begin to do the things necessary to divide the cell.
Which brings us then to metaphase. So once those things are complete, we would say that we are now finished with prophase. We're moving into metaphase. The prefix meta means middle. That can give you a hint as to what is going to happen in this phase, and you can see that illustrated already. The first thing that we're going to have happen is that the centrioles will finish their migration, so at this point they have officially arrived at opposite ends or at poles. Next, you will see the spindle fibers growing out and actually attaching at the centromere, so that middle point that we illustrated last lecture that you can actually see again in this picture on the left. Sort of the hips of the chromosome, if that's a good visual for you. They will attach there, and the chromosomes will line up in the center of the cell, which is referred to as the metaphase plate.
Right. We also will, once the metaphase plate or the chromosomes are lined up at this kind of fictitious equator, if you will, there's no physical structure making up that band that I just drew in, but that is referred to as the metaphase plate. And once the chromosomes are lined up at the metaphase plate and the spindle fibers have completely set or completely attached themselves and moved the chromosomes to that plate, we refer to that as the spindle apparatus, and it is complete at this point. So chromosomes lined up along the middle, centrioles are going to be in the poles, the spindles are produced, and it's important to distinguish between the spindles that are actually attached to the centromere and the spindles that are being produced that are not going to attach themselves to the centromere. So you'll notice that there are more spindle fibers than we have chromosomes. And so if we were to progress this further, these spindle fibers would actually bypass the chromosomes and would actually connect from centriole to centriole, which is going to be vital as we kind of progress through this M-phase.
So next we have what's called anaphase. The prefix ana means apart. Again, giving you a clue as to what is going to happen in this phase. The spindle fibers begin to retract back toward opposite poles. So they are moving away from each other. They have reached out and grasped these chromosomes, and now they are pulling apart. And so you can see in the diagram that at this point the chromosomes have been split down the middle.
And so the sister chromatids have been separated. This is in the last video when we talked about when we refer to these again as chromosomes. So they would have been sister chromatids together. They are now individual chromosomes which are unduplicated. So it is important to distinguish that. These chromosomes right now are unduplicated individual chromosomes. And that's important because we want to make sure that our cell doesn't actually change its ploidy, which is a term we won't typically get to until we get to genetics, but cells have to produce identical cells, and diploid cells have to remain diploid. That's one way that they do that.
Also, at this point in time, the cell, as you can see illustrated, is sort of elongating. It's going to grow outward, becoming a little bit more oval in shape. That's going to help us pinch it in half in the future.
Yeah, and again, just like in the last kind of picture, these spindles did not actually showcase the completeness, I guess, but these spindles would actually be complete, or they would be consistently from centriole to centriole, and these are the spindles that are actually pushing this way, which is causing the cell to elongate in opposite ways, or in opposite poles. So these spindles that are attached to the centrioles are going to be pulling into the poles, and the spindles that are pushing from centriole to centriole will actually be pushing in the opposite direction.
And that brings us to telophase or telophase, depending on who you ask. And in this phase, I like to picture this phase as the reverse of prophase, just with two nuclei. And so we'll talk about some of the things that are going to happen in telophase.
So if you remember in prophase, the first thing we talked about was the nuclear envelope dissolving. Well, now since we've divided the cell's nucleus, we have to remake the nuclear envelope. And so in this case, or in this phase, the nuclear envelope is going to reform around the chromosomes. So again, it's a dashed line. It is reforming. It is not fully set, but it is being set or being produced. The chromosomes are going to be basically going inactive or returning to the inactive form. So the chromosomes, or those individual X shapes that we separated and moved to the middle, are going to be decondensing back into the inactive form, which is the chromatin. So in this case, they're going to be returning to those individual kind of dots.
The spindle fibers, as you are witnessing here, begin to break down and dissolve because they have completed their function of elongating the cell and pulling apart the chromosomes.
Yep, and no longer need to actually attach or manipulate the chromosomes or move them around. They're already in the nuclear envelopes, and those nuclear envelopes forming the new nuclei. And not showcased very well in this diagram, but there will be a cleavage furrow that forms, which is pinching in the cell at that metaphase plate, or what was the metaphase plate. And so if you were to progress this phase through telophase, as you end up, the cell actually will begin to start pinching in. And it's this pinching in that is going to cause cytokinesis to take place.
So, like I realized there that we haven't actually told you what the prefix telo means or telo means. We skipped that part at the beginning, but telo means final or end. If that's helping you kind of remember these phases, that this is the last phase of mitosis. Notice at this point, we still don't have two cells, but we do have two complete nuclei by the end of this which will move us into cytokinesis.
Right, and remember, that is the definition of mitosis, or the mitotic phase, is we are not dividing the cell, we are dividing the nucleus. It isn't until cytokinesis actually causes these cleavage furrow to come together and ending cell division, or what we refer to as cell division, and ending up with 2 new daughter cells. So what are the things that are happening during cytokinesis? Again, in this diagram, you can see that cleavage furrow forming. This pinching in of the cell membrane is going to create the 2 new daughter cells. Notice that in this diagram also, telophase has completely finished, so the nuclear envelopes are intact. The DNA is in the form of the inactive form chromatin. You don't have any more spindle fibers, so these cells are ready to basically begin their new life as a cell, whatever the type of cell it is. And so the events that happen during cytokinesis would be that cleavage furrow forming, or eventually fusing the 2 cell membranes, completely separating the new daughter cells from each other.
Which is then going to officially complete the cell division process. So through cytokinesis and mitosis, we have completely divided the cell.
It forms 2 new daughter cells.
And it is a little bit different in plants and in animal cells, because plant cells form the cell plate, which ends up being the cell wall. They have a little bit more rigid cell wall around the outside that animal cells don't have, and so they have to build that, and so it's a little bit different in plant cells.
Yeah, and one thing to kind of, since we've been focusing on prefixes, cyto means cell and kinesis means split, and so this, if you can remember the word descriptors, splitting the cell refers to splitting the actual cytoplasm. So when we look at the cytokinesis events that happen in animal cells and plant cells, remember these are both eukaryotic cells, and so that's a good distinction. Mitosis is something that happens in eukaryotic cells because mitosis is dividing the nucleus, and so we need a cell that has a nucleus. That would be a eukaryotic cell. Animal cells and plant cells would be the 2 different types of eukaryotic cells that we're going to focus on in this particular lecture. Animal cells are cells that make up all animals, including us. Plant cells are going to be cells that obviously make up plants, and like Mr. Schmidt said, they contain the cell wall. And so if we look at animal cell cytokinesis, at this metaphase plate, there will be this thing called a microfilament ring. Now, microfilaments you might remember from our cell cytoskeleton talk. Microfilaments are a microskeleton component that are made up of protein, and there's this contractile ring that forms around kind of this equator where the cell will actually divide. It constricts, and that's what forms that cleavage furrow, eventually causing the cell membrane to come together until the 2 new daughter cells kind of snap apart. That's the easiest form of cytokinesis because we're just pinching in and snapping the 2 new cells apart. In plant cells are a little different because they have to physically build the cell wall, which is referred to as a cell plate down the middle. And so they have to construct this cell wall, which then is eventually joined in the middle with the cell membrane, because they do still have a cell membrane, to form 2 daughter cells. You notice that set of daughter cells is much smaller than the initial cell, which is why they have to also go through interphase to grow and change after that point in time. Right, and that's in both of these cases, this is how animals and organisms in general are going to be growing, right? So as a plant is growing, you're taking one plant cell, maybe it's stem, maybe it's leaf, but one cell, and you're going to divide it into 2, and then those 2 new daughter cells will eventually start growing outward, becoming the same size as the original cell, and that's what grows the plant overall, not only in width, but also in height, right? So a plant will continue to grow out and will grow higher, just like you do.
That's all we've got for you today with mitosis and cytokinesis.
See ya.